Thank you very much for the kind introduction, and it's a pleasure to be here. When Amos asked me to talk about neuropathology, I realized that one of the biggest changes, besides moving this up, okay, these are my disclosures. They are not important for this talk. One of the biggest change in talking about neuropathology is really to define what we do. We are not very good as a group to communicate about it. And I learned very well this lesson when years ago I was contacted by a newspaper in Brazil saying they wanted to report about the brain bank we had started 1 year ago. So they came. I was very happy, thinking they wanted to hear what I had to say. But actually, they were pretty much looking for the jars. The jars. So all the time he kept asking, where are the jars? So I realized that what they expected to see was a collection of this kind of jars with full brains inside. And they thought the neuropathologists were people with X-ray, and we would look through these full brains, and we would tell everything that was inside. So it was a very big lesson for me. But now it became much better, because some months ago, this movie about the history of chronic traumatic encephalopathy came to the movies, and it went very well in the box tickets. And so this movie, for those who saw, know that it's about the history of a neuropathologist, Dr. Omalu, who seems to identify CTE in football players. And in most of the movie, whatever he was criticized or praised for what he was doing, the actor would say, you know, I'm just a neuropathologist. So I'm sure that this movie helped to put faces into neuropathologists, and also to explain to people that actually we don't have jars, we have to section the brains. And this is what we have, Tupperwares full of brain slices. And actually this movie showed that we have better microscopes that Cajal had when he started to look at the brain. Helps a lot. So today I will talk about beyond the microscope in neuropathology and all the technology advances and changes that happened in the last years that had or will probably have very soon impact in the clinical practice for all of us. And I think the first question we ask when we think about neuropathology and human brain, we are specialists here. We know everything about the cascades and neuronal laws and all these problems. But I will ask you, do you know how many neurons make the human brain? These very fundamental questions. Could you please raise your hand if you think that it's less than 100 millions? Less than 1 billion? Less than 10? Less than 10 billions? Very good. So if you look online, if you Google it, you will go to the Neuroscience for Kids, and they will give you this very magic number of 100 billion neurons in the brain. But actually, if you look for literature about it, you will see that this is really a magic number. There is not a lot of fundament about it. And thinking on this, we will think, so how it's possible to measure how many neurons we have in the brain? This is a human brain. It has about 18 centimeters in the longer axis. And when we cut it, we will see all these dots over here, and these represent neurons. And as you see, they are not distributed in a homogeneous way. So in order to figure out how many neurons we have in the brain, actually we will have to look at piece per piece. And this is a very challenging task. Luckily, about 10 years ago, these two researchers from Rio de Janeiro, Roberto Lenti and Susana Ruzel, they had this very bright idea of transforming this full brain in what we call brain juice. So pretty much they would take pieces of brain and use this spotter to homogenate it. It will destroy the brain, the neurons' membranes, but it will preserve the nuclear membranes. Then they use a camera of Neubauer and immunocytochemistry, immunofluorescence, to stain the cells with markers for neurons. Is a marker called NeuN, and a marker that will stain the nucleus of every cell that's called DAPI. So looking at the camera of Neubauer, they now transform the brain that has a very heterogeneous distributions of neurons in a homogeneous distribution of neurons, and they counted how many neurons there are in the brain. Actually, this was the proposal. And indeed, we did it with a brain from the brain bank in Brazil, and we realized that The 100 billion numbers was not so wrong, and actually what we found was 86,000 neurons. So this was interesting information, not 100 but 86. But what was really puzzle for me is that despite the fact that the cerebral cortex corresponds to about 80% of the brain mass, half of the neurons we have are in the cerebellum. And this was the most intriguing thing for me, especially because, you know, in many of the diseases we study, we try to either consider the cerebellum as a one thing or overstate it altogether. So why do we have so many neurons in the cerebellum? Of course, now we learn more about the functions of the cerebellum, even some functions related to cognition and so on. But it was the most interesting story for me. And we use this method to answer some other questions that I find interesting to share with you. So for instance, okay, we look at the whole brain. So let's compare what happens between men and women and what happens as we age. So this is one of our papers that was published last year. So we look at this medial temporal lobe, the hippocampus, together with entorhinal cortex, amygdala, and it became clear here, examined several number of people, that men have more neurons and more non-neuron cells in this area than women. And you know, it also intrigued me, and I asked my husband how it's possible that despite he having so many more cells than I have to remember him, that he has to take the trash out every week. And he answered that, you know, he used all these extra neurons to remember the name of all the characters of Lord of the Rings because he knows all of them. So maybe this is the reason for this. And then we look at the difference along age, and what we found out is that we didn't find any difference in neuronal number of cells altogether, but these are regression lines. But it seems that number of neurons peak around the age of 70, and then they go down a little bit, but not much less than what we had when we were 20s. Anyway, so there were many women in our group, and we decided to take revenge on this. So we look at the olfactory bulb. And finally, we found a place in which women has many more number of neurons than men. As you can see here, the number more neurons, more number of no neurons, and total number. But if you look at the mass, and this is important information because usually when we use imaging methods, we are looking at mass and volume. We didn't find any difference, as we didn't find difference for other area as well. So we decided to take this method for looking at a number of cells and comparing men and women to look at problems that intrigue us in, for instance, neurodegenerative diseases. So in this study, it was published in 2013, we took 5 brains of people who were controls, meaning no cognitive decline, no substantial pathology in the brain, and we compare with other people that didn't have any cognitive decline, but they met criteria for severe Alzheimer's disease. And finally, we compare with a third group of people that had severe dementia, and they did have also Alzheimer's disease in the brain. So actually, if you compare group 2 with group 3, they have the same amount of protein, plaques, and tangles. So we look at the cerebellum, and we look at the medial temporal cortex again, and we didn't find any changes in the cerebellum. But looking at the medial temporal cortex, we noticed that despite the fact that group 2, here in gray, and group 3, here in black, had the same amount of plaques and tangles, the number of neurons of group 2, no symptoms, really compare with the controls and not with people with symptoms. And it just came to corroborate previous observations and investigations done in a slightly different way, that indeed the best correlate for cognitive decline is loss of neurons, most likely. Or maybe, I mean, we are not really measuring loss. Maybe these people had more neurons to start with, but maybe the number of neurons that are there and they are functioning well. And that you can have in a in a significant number of people, and we do see it in neuropathological examination. They have a lot of protein inclusions like plaque and tangles in Alzheimer's disease, and it had no clinical consequence at all. Okay, so can we look exactly why some neurons or why some people will be vulnerable to lose neurons probably when they have this accumulation of proteins and it will have not? So when we look at the literature, we find very interesting work done with animal. For instance, this is called brain ball as opposed to rainbow. What these researchers did, they injected some glowing substance in mice, or they transfected the genes. So when they look at the cells of the hippocampus, they glow in different colors depending on these functions. And we have to tell, I would love to look at the brain of men versus women to understand exactly what are the cells we need to store dubious information, like the Lord of the Ring characters. But we are not there yet. We cannot do this with humans. But there are some other things that can help us a lot. So one of these methods is called laser capture microdissection. This was a method that has been described about 20 years ago. Of course, it evolved since then. But pretty much what it allows us to do is to select areas of the brain of interest and dissect them out, like this is a glioblastoma, and then examine these cells either using special stainings or genomics, proteomics, and really try to learn what is the difference between cells that have something of interest as compared to normal cells. And this is just to exemplify how it's done. So we take a piece of brain, we freeze it, cutting in very thin sections, and then we have this special microscope linked to a computer. We can see the cells, we can even stain the cells beforehand, and with a joystick we trace the cells that it's captured in one tube, and then we can use it for different exams. That's very popular in the lab. All the students, they are burned out and need occupational therapy, are given the opportunity to spend some days here with the joystick, and this is how it looks like. So doing that, we can do, for instance, proteomics assessments like we did here, and this is pretty much, we try to figure out how many proteins we identify and how they are different between one kind of cell from the other kind of cell. But even the way we look at these proteins or genes we find in a cell, the methods for doing this improve a lot in the last years, and we are learning a lot about it. So I'm showing here an example of a method that's called complex network driven, and we use this to examine cells in the dorsal-motor vagus nucleus, locus coeruleus, and substantia nigra, who are important nuclei affected in early Parkinson's disease, and compare this to controls. So pretty much we dissected cells in these areas, and instead of just, listing the number of proteins, with this method that it's based on social media, we could see how each one of these transcripts of these genes are correlated to each other. So based also in social media, they call these genes that are interconnected or that had certain kind of connectivity with other genes, more than 10, they are called hub. And there are those who have less than 10, but they are connected to a lot of hubs. We call them VIP. And they are very important. We are learning as we see controls versus diseases. So for this example here, this is the dorsal vagus. We are seeing the figure in controls, and as you see, it changes a lot in Parkinson's disease. But one thing that was very intriguing from this work is that we realized that when we look at the vagus, for instance, most of the genes that were important in this network, they were protective. They had protective functions like against oxidative stress, regulate lipid metabolism. And even in the Parkinson's disease cases, we saw that the genes that became more important, they are also related to protection. For instance, neuronal plasticity or cell renewal of stem cells. And actually, the picture for the locus coeruleus was about the same. A little bit different genes, but related to antioxidant properties or neurogenesis. And in case of Parkinson's disease, the same thing, as if in Parkinson's disease, these genes will be, or the functions of the cell will be changed to try to really protect against whatever injury was going on there. But then the picture changes in substantia nigra. Again, of course, the picture of controls and PD changes a lot. But the genes that we found different expressed in case of the substantia nigra was pretty much related to neuroinflammation, protein folding, as if at this point the brain would give up and you really succumb to the pathological process. Of course, this is cross-sectional, this is very initial, but it's just to illustrate the power of this kind of technique, especially when we have very well characterized human tissue and we can correlate these results with results of neuropathology and clinical phenotype. And this idea of using RNA or looking at different areas of the brain, it's really becoming very standard, and probably you've heard about the Allen Brain Human Atlas, that they dissect several areas of the brain, almost all of them, and they are profiling RNA so we can have a database for controls and we can compare our results. So all this is very good, but we all do this with patients that already died. And actually we are interested in trying to learn about the disease or even normal controls as the people are alive. So how can we achieve it? And this is what I will try to show in the next few slides. So this is just to illustrate. This is a 7 Tesla MRI. It's the only one we have in Latin America so far. And this is installed in the autopsy service of the University of São Paulo. We use it for research mainly, in vivo and ex vivo. And this is Edson Amaro. He's the radiologist responsible for this. He's here in the back. And it's a very nice project we started last year. And mapping the brain is not something new, and it's not something that we try to do only now that we have neuroimaging available. But it's done for over 100 years. Probably many of you heard about von Economou. He, from 1912 to 1925, he almost alone devised a way to dissect the brain in several areas, and he described 107 cytotectonic areas. At that time, especially, they felt that correlating function with structure was very important, and it is. We still use it for many applications today. And with these observations, not only he was able to look at these different areas, but he was able to identify new kind of neurons. This is a good example. It's called von Economou neurons. It's present just in few areas of our brain, and it has to do most likely with the ability of us to recognize ourselves. So only animals that can recognize themselves in the mirror as themselves, for instance, have these kind of neurons like elephants and whales and apes. And these neurons are interesting, and this is a work from Bill Seeley, because in behavioral variant frontotemporal dementia, these are one of the first neurons to degenerate, regardless of the molecular signature of that's giving the disease, either peak disease, that's tau, or FTLDU, that now we know it's TDP. As in Alzheimer's disease in controls, nothing happens to this brain. So to understand the structure of the brain, and even to this level, what kind of neurons might be vulnerable to certain diseases, might help us to maybe create strategies to protect these neurons regardless of whatever the protein is going on there. Von Economou took this 13 years to describe the cytotectonic areas, doing a very painstaking work. So he will cut the brain, this is one example here, and then he will measure how thick was each layer of the brain. This is very nice, but it took him 12 years to get to there. Nowadays, I think we can do better, and we think we do better with neuroimaging. This is a 3 Tesla MRI. And as you see, if you compare with gross anatomy, looks pretty good, but we still have the challenge of resolution. So we can look at single neurons under the microscope, but we are still not there in the, when we have MRI, even with 7 Tesla. And probably we will feel that improving the MRI machine, going to higher fields, will help us. It might, but we have also to consider that there are limitations of our tolerance against magnets. So probably we need some other creative ways to get there. So pretty much what we are trying to do is to approximate the resolution of MRI. This is the Earth seen at the same resolution that Pluto was seen until the latest probe got there. And this is so you can see how much information we are missing for not having the right resolution. So how can we put them together? A good way to do it will be to really compare what we see in post-mortem tissue with what we see in neuroimaging, trying to create maybe a matrix for comparisons. The problem is, in order to compare things, they have to be at the same space, at the same coordinates. And as we don't have this X-ray vision to look at the brain, we are forced to section the brain really to get information from this. So I want to illustrate this problem with an apple. Here's an apple. Imagine this is the brain. We have to section to look at the brain. Seems pretty easy, but because we have to preserve the brain and prevent that it will rot, and we have to fix in formalin or some other things, it really deforms the brain, and this is what we have, and it creates a big problem when we try to do correlations. So we are trying to fix it. This is one way we do in our group, but certainly there are other ways. So pretty much, because we have a lot of autopsies in Brazil, we have the opportunity to do ex vivo, but still in situ before the autopsy MRIs, and then we are very creative and we print in a 3D printers a model of the patient's skull based on neuroimaging. We fix the brain right here, and then we embed the whole brain and we cut in very big machines. And doing this, we can minimize the distortion that will help us to try to do this correlation. And this is a good example, very minimal distortion. So we use this already for several clinical applications. I will show some of them. This is the isodendritic core. This is a series of nuclei, mostly in the brainstem, but also there is this in the telencephalus. And it's very important for Parkinson's disease and Alzheimer's disease because they produce, they are monoaminergic and they are cholinergic. And this is the nucleobasalis of Minert. So we know that the nucleobasalis of Minert shows pathology and changes in Alzheimer's disease very early, years before the symptoms. So what we did, we used one of these brains that we had, imaging and pathology. We reconstruct the nucleobasalis of Minert. And then Stefan Teipel, he used this mask and he used VBM to create a mask in which you will be able to identify the nucleobasalis of Minert in MRI, even in 1.5 Tesla. This is what he used on his first studies. I'm showing one of them. This is a multi-center study in Europe in which he compared 150 healthy controls with AD patients, and he showed that the area under the curve looking at one part of the nucleobasalis, CH4P, was pretty much similar with the results we find for the hippocampus. So he decided to move forward and look, how about if he compare controls to MCI patients? This is in blue, the MCI patients that corresponds to this CH4 area, and it seems to be a prominent technique that we might be able to use to monitor changes in the future. We also use this technique to improve deep brain stimulation. This is a work led by Eduardo Alho, a neurosurgeon in Brazil. So what he did, he reconstructed subthalamic structures correlated to MRI. He did it with hypothalamus too. There is a very big interest in the posterior hypothalamus that's difficult to see but has important for cluster headaches. And now we are working with companies that do these navigators for stereotactic surgery to really embed this information in their navigators. This was the first problem. It was resolved, but this was done with small pieces of the brain. So how about if you want to look at the whole brain and go really high throughput? And then we have a second problem, that is our computational capabilities. It was well illustrated by this paper 2013 from Katrin Amus group, in which they try to to digitalize a whole brain in a very good resolution, 20 micrometers, and try to predict, for predicting, for instance, site architectonics. And what they show here, that even using the best algorithms, what the maximum they could do is to reduce this data to 1 terabyte. Can you imagine 1 terabyte of data per brain to analyze? But as you can see here, they couldn't take care of the distortion. I mean, this was not the question in this brain, but they could reduce to 1 terabyte, and it was so important they published in Science. So we decided to take the challenge also in our group. So just to give an idea of scale, MRI data set of one brain at 1 millimeter cubic resolution gives about 10 megabytes of data. With our way to process the brain, that it's a little bit produce less data than what Catherine Amos did, we get 80 gigabytes of data. So we started to work with these 2 data sets for doing these reconstructions and comparisons. And I have to tell that we exhausted all the computational capabilities of UCSF, that it's good. We used the best clusters there. It was not enough for us. So luckily, we are in the Silicon Valley, and we are also very close to the Lawrence Berkeley National Lab. They have supercomputers. So we are collaborating with them to really be able to analyze our data. So the first thing we did, instead of using traditional computing, we used something that's called high-performance computing. That's pretty much divide our data in different data sets. They are processed in parallel, and then they are put together. And this is what we got so far. So this is a 3D reconstruction of the brain in our MRI data. As you can see, there is no distortion here, and the correlation, it's pretty good. Of course, we calculate the error. This is a paper that we submitted last week. And the resolution we could obtain in 3D was 15 millimeters, which we think it's pretty good. And, oops, to illustrate what we get, we have a movie here. So our hope for the future is that we now can take this data, that it's co-registered at a good resolution, and create algorithms to examine each point of these correlations and create a library that we can then incorporate in the way we interpret imaging. And this is for structural matters or for other matters as well, as I will show so far. So we can use it not only for looking at site architectonics, but also to look at the integrity of the white matter. This is a very clever method invented by Huberto Axer in Germany. So what he does is he uses polarized light to try to understand the microstructure of the white matter and compare, for instance, with DTI or other modern acquisitions techniques that has to do with the white matter. To illustrate it, this is the polarizer apparatus. This we have in my lab. We collaborate with him. I have to tell that technology also helps us a lot. He spent 30,000 euros to build his apparatus, and I spent 2,000 euros. And the difference is because we could print everything in 3D. He didn't have at that time. So now we just design it and print it and install, and it makes our life much easier. One of the questions we are studying is the perforant path. You can see here the hippocampus and entorhinal cortex. This is how it shows in polarizing light. And this figure is to illustrate that in the size of one voxel that we obtain in clinical DTI, 2 millimeters, this is all the amount of information we have with histology. So really trying to do the correlations can improve the way we can interpret this imaging. So the cake on the cherry on the cake is, how about molecular imaging? Can we really predict which kind of protein depositions we have in the brain just looking at imaging? There are several groups working on this, including us. I think we can solve already the histology part. We can reconstruct in 3D protein 3 of whatever disease. This is a parametric map of dipeptide inclusions in the cerebellum in a C9 patient. And the biggest bottleneck in these studies is that we depend on volunteers that come from hospice care, because in order to do this correlation, the gap between imaging and pathology have to be short. So it's important that we try to minimize as soon as possible. And as you know, hospice care patients, they also have a lot of challenge that even prevent imaging, even if they are willing to volunteer, and many are. So to conclude, I will just take this word from Ramon and Cajal. The brain is a world consisting of a number of unexplored continents. I think it still are now incorporating technology to what we are doing, especially to well-characterized brains. I think we will be able to answer a lot of questions. I don't know how much it will lead us to answer or to treatment of all the diseases, but certainly I think we will see a lot of changes in the next years. I would like to finish just thanking all the team working on this. The pathologist doesn't work at all alone anymore in our labs, buried under a microscope. This is my team at UCSF, but there are a lot of people participating in this work, and I would like to thank you very much.
Thank you for this very interesting conference. Are there any questions? Yes.
Congratulations. It was a beautiful presentation. One of the questions that we have as clinical neurologists is when we perform a SPECT in a patient with Alzheimer's, we usually see the same areas with hypoperfusion. And you actually mentioned that at the beginning. My question, what do you think? The hypoperfusion is the reason or the result of the atrophy of neurons? I know it's not an easy question, but I think you have more elements with all the information you already have that maybe you can give us your concept.
I think this is a very fundamental question. I'm not sure if I have the answer. Unfortunately, one of the limitations of neuropathology, we are not seeing function there. So I can see that neurons are not, maybe they are shrunken, but I don't know how they are really functioning in life. So I hope that improving the interpretation of in vivo studies with this correlation work, we will be able to answer this question. But whatever answer I will give you, it will be a guess.
Usually in pathology, we distinguish, or we try to distinguish between apoptosis, like a process, or necrosis of the cells. Can you help us in Alzheimer's? What is more common in Europe? Or no?
Yeah, so in Alzheimer's, what we have is programmed cell death. Apoptosis is one kind of programmed cell death. It's different from necrosis that's usually driven by lack of oxygen, or it's a different process and it comes with inflammation. But apoptosis is not the only programmed cell death we see in Alzheimer's disease and only in the other neurodegenerative diseases. There are some cell death mechanisms involved that are what we call caspase independent. Caspase is a very important element of the classical apoptosis. There are issues with autophagy that's not necessarily a cell death mechanism, but apparently problems with autophagy will trigger problems related to cell death. So, but it's not, I would say, I think it's fair to say it's not necrosis, but it's programmed cell deaths, different types. We don't know exactly which ones are the most important in early stages of the disease yet.
Thank you.